Hello everyone. So in today's video, I'm going to explain the regulation of purine synthesis. This was supposed to be a part of the previous video that I posted, but then I forgot to add this to that video. So yeah, let's see. So I hope you all watched the previous video that I posted, which is the pathway for purine synthesis. So the first step that we need is ribose 5-phosphate or alpha D ribose 5-phosphate is converted to PRPP. I'm going to write down the steps first and then tell you what's inhibiting them. So basically this pathway undergoes a feedback regulation system. So let's see. PRPP is further converted to 5-phosphoribosylamine. This is the irreversible step or the committed step in this reaction. The enzyme used to convert ribose 5-phosphate to PRPP is PRPP synthetase. The enzyme used to convert PRPP to 5-phosphoribosylamine is glutamine PRPP amidotransferase. 5-phosphoribosylamine undergoes 9 steps in order to form IMP or inosine monophosphate or inosinate or inosinic acid. From IMP, it is split into two pathways. In order to form guanylate and adenylate. So let's see what those are. First, it forms adenylosuccinate. Using the enzyme adenylosuccinate synthetase. And on the right side, it forms xanthylate. Using the enzyme IMP dehydrogenase. Since the enzyme is a dehydrogenase, we know that NADH is released here. Final step. This gives us adenylate or AMP. This gives us guanylate or GMP. Enzyme used here is adenylosuccinate lyase. Lyase is used in order to release fumarate from adenylosuccinate. And here the enzyme used is XMP or xanthylate, glutamine, amidotransferase. Then we can further write ADP and ATP. Same here as well. GDP, GTP. So AMP, ADP and ATP inhibit the step which converts PRPP to 5-phosphoribosylamine. So I'm going to draw that. Same on the right side. GMP, GDP and GTP inhibit that same step. Minus sign or X signifies that the reaction is being inhibited. Another inhibiting step is AMP inhibits adenylosuccinate. GMP also inhibits that same step. By the same step, I meant the same step on the right side. One more inhibition occurs here. It is done by ADP. So I'm going to mark that in blue. This AMP can be converted into ADP, and that's what inhibits this enzyme. Now let's talk about activators. So when it comes to activators S2, ATP activates this reaction, XMB glutamine aminotransferase, and GTP activates the adenylosuccinate synthase reaction. So there you have it. That's the regulation of purine synthesis. The text that I'm using to refer is Lineager 7th edition. So if you guys have any doubts, you can refer the text or you can ask me questions in the comments. So yeah, that'll be all. Thank you. Have a nice day.